Welcome to our Lunch, Learn, Link seminar today. We host seminars a couple times a month during the academic year, so I hope that you all are getting emails about the event and can put them on your calendar. This year, we're not able to stick with the same day and the same time in the same room, so bear with us. We're moving from one room to another or from one day of the week to another. And I know, that keeps us from thinking that an event is coming up, but we will have the events and hopefully, the change in the rooms and difficulty having a routine won't keep you from coming. We welcome our online watchers of our seminar series, so a special shout out to the Department of Health and Mental Hygiene, the University of Maryland, and Howard University in particular. And I'll just say this seminar is sponsored by the Cigarette Restitution Fund and by the Sidney Kimmel Comprehensive Cancer Center. So we hope this generates some thoughts on your part, maybe some partnerships, some recontact after the seminar. It's my pleasure to introduce Ben Ho Park today. Ben actually was a Cigarette Restitution Fund awardee years ago and is a medical oncologist at the Cancer Center today, specializing in breast cancer care. So he has some new information about genomics in breast cancer and then some innovations in breast cancer care. So I welcome Ben.
Thanks very much, Norma. It was funny. On the way over, I just realized that the first time I met you was actually because my first grant on faculty was actually through the Cigarette Restitution Fund, and Norma was actually the PI. I believe you were the PI, and still is. So it makes me really feel old, for one thing. I've really grown up here at Hopkins, and a testament to that is I didn't need these when I first joined the faculty 13 years ago, but now I clearly do, so bear with me, please. The title of my talk is Advances in Genomics in Breast Cancer Care. Actually, now that I think about it, I can just do this. And part of this, I wasn't entirely clear what type of audience that I was going to be addressing this to, but I've had some lectures here in the School of Public Health before, and I know the range is quite wide, from people who are sitting in on courses and maybe still at the undergraduate level versus all the way to full professors. And so I'm going to try to actually distill this down, and so forgive me if this seems a little bit too simplistic at times, but hopefully I can walk you through the process of how, at least in the breast cancer field and in oncology in general, that we've really tried to embrace new genetic genomic analysis for how that could impact patient care. These are my disclosures. I sit on a number of SABs as well as receive royalties and research agreements from various entities. And really the objective today is really to describe the different technologies that we use for genomic and genetic analysis of breast cancers, and how some of these now are actually moving their way into the clinics and some already have. As a quick overview of what I want to talk in the next probably 45 minutes is, first, what are the analytes that we actually measure for these omic tests? There's DNA, RNA, and protein, and I'm going to really focus more on DNA and RNA, but I am going to spend a bit of time on RNA because that's the one area right now, clinically, at least in the United States, where we do have real validated tests that can affect our management of breast cancer patients. And then a little bit more in the future, including what we've started here at Hopkins called the Gateway Tumor Board, in quotes. I'll explain what that is at the end of the talk, but really how DNA is moving forward, and I think will actually offer us the ability to manage patient care based upon the genetic genomic profile of a patient's tumor. All right, so again, forgive me if this is a little bit too simplistic, but I think probably everyone knows that DNA is really the cellular blueprint of all living things, and that there are 4 kind of letters in the alphabet, or base pairs, A, C, G, and T. That really the central dogma, which I'll present on the next slide, is DNA goes to RNA, which goes to protein, and there's very few exceptions to that, though there are some. And in general, each cell of the body that carries DNA will usually have 2 copies, one from mom and one from dad. This again is the central dogma that I just said in the preceding slide, so that I think we all learned in high school biology now that DNA is a double-stranded entity, and that it is really the basis for transcription, which leads to then RNA or messenger RNA. And this gets translated outside the nucleus into proteins, which ultimately is kind of the business end of what DNA is really going to do in terms of the cell and cellular function. So this isn't a talk about proteomics, but it's kind of funny, we're working backwards. We actually really use more protein these days in clinical tests for breast cancer. We're starting to use a lot more RNA, as I alluded to, and then eventually DNA. And so the proteins that we typically use for breast cancer management is really at the level of immunohistochemical staining. And so for breast cancer, I think many of you know, it's not just one disease, it's actually many diseases, but the three receptors that we really look for or assess, at least in a pathology slide, are estrogen receptor, progesterone receptor, and HER2. In some institutions, including Johns Hopkins, we also use a protein stain for Ki67, which is a proliferation marker. This is not standard of care, but more and more pathology labs across the world are really adopting this. And these markers have both what we would call prognostic and predictive utility. Prognosis in our terms clinically usually means what is the likelihood the disease is going to be back in 5 or 10 years, or the patient may be alive or not alive in 5 or 10 years. Prediction is really something very specific in clinical oncology, and we tend to think of predictive markers as something that will actually give us the likelihood or not of a given therapy being effective. And there are other tumor markers that are in use for breast cancer right now that are protein markers, specifically the MUC1 antigen. This is something that's secreted by most cancer cells, and there are a couple different protein tests that really antibodies against the MUC1 antigen that can be used clinically. When these were tested about 2 decades ago, however, they weren't really that useful. And so for early stage disease, we actually don't use them, or at least our guidelines suggest not to use them, and they're optional for metastatic disease. Part of this is that they're not specific enough, and they're not sensitive enough to catch disease at an earlier state where it can actually affect clinical outcome. Moving on to RNA, again, I think most people in the audience know that RNA is not a 1-to-1 ratio with DNA, that a given DNA gene can actually encode many RNA molecules, and this is kind of the term that we now colloquially call gene expression. RNA itself is a very labile molecule. It's single-stranded, and because of that, it can degrade very quickly. And so there's been a lot of investment over the past 2 decades of how to actually preserve RNA and achieve the best high-quality analyte as possible. Typically, the RNA will get converted in the lab back to cDNA using a molecule called reverse transcriptase. And the challenge in the past was, well, how do we measure all the 30,000 or so different RNA species in the human transcriptome and actually then make sense of this and apply it to clinical utility? There are a number of ways, again, for those of you in the lab already know this, how to measure RNA. A northern blot is the classic. This is something that's rarely done anymore, but when I was a graduate student and even a postdoc, this was the norm. And so I'm not really going to get into what that is, but suffice it to say, it wasn't a very quantitative or sensitive assay by today's standards. Bert Vogelstein's group in the mid-90s published a seminal paper now called Serial Analysis of Gene Expression, or SAGE. This was really the first attempt at an unbiased global gene expression assay. It had its limitations. It was very cumbersome and expensive at the time to do, and it's pretty much fallen out of favor because of newer technologies. But back in the day, this was really heralded as something very visionary and very powerful and important tool for analyzing gene expression. Along came quantitative real-time PCR, and again, I think most people in the audience know what this is, so I won't belabor this. Microarrays really were the first, I think, platform or technology to take off to allow us to do global gene expressions. And these were originally on chips and then on beads. It really allowed for a high throughput way to query for the human transcriptome. The limitation in the early days, however, is that we didn't know the whole human transcriptome, so it was biased with just the genes that we knew of, and we could lay them down on the chip or bead. And then today, of course, RNA-seq, which I'll mention a little bit later, really falls into the realm of next-gen sequencing. And so again, RNA for gene expression assays. So for the first use really was to just do global gene expression profiling in the lab and using some of the various technologies that I mentioned. There was kind of a head-to-head competition in the 90s between these unbiased SAGE approach versus microarrays. And I think ultimately because more and more of the genome became sequenced, the microarrays became cheaper and faster to do and sort of won out. With better biostatistics and bioinformatics, researchers use these global gene expression categories to really start thinking about how do we subgroup different types of cancers. And Chuck Peru, along with David Botstein at Stanford, really was the first group to take real primary breast tumors. So these weren't cell lines, these weren't mouse models, but actual patient tumors, and subject them to a global gene expression array analysis. So you may have heard these terms already, but this is where it originated from, the so-called luminal A's, luminal B's, HER2, and basal. This is all from the early days of using microexpression arrays and looking at hundreds of tumors and categorizing them by their unique gene expression signatures. There were limitations. This really required frozen tissue in the past, and back then it was actually fairly expensive. SAGE, again, was one of the first lab-based assays for global gene expression analysis, and this was really ingenious at the time because the idea here, we didn't have next-gen sequencing, so it was standard sequencing, which was expensive and difficult to do. How does one actually then, therefore, query the whole genome in an unbiased way? And so Ken Kinsler and Victor Valcalesco and Bert Vogelstein came up with this idea that if we could make little tags with an anchoring restriction enzyme site at the 3' end of a given transcript, that would be enough information content to uniquely identify that tag in amongst the 30 or 50,000 transcripts in the human transcriptome. The key was that if you could make these short and then you can serially concatenarize them, you could put this all in one plasmid, and then rather than one sequencing run only having a single read for a single transcript, now you could get dozens, maybe sometimes up to 100 different tags. Now you've increased your throughput by several fold, and that allows one to put it into a computer and then do some qualitative and quantitative analysis. And again, for the time, this was really groundbreaking, and this was still used for several years in the Vogelstein lab. Certainly when I was a postdoc there, this was kind of all the rage. But then along came better technologies, and again, with the sequencing of the transcriptome, microarrays became more and more common and popular. They did originally have this disadvantage of biased expressions because we could only put down what we knew in terms of the transcriptome. And they also did require large amounts of input DNA, usually from fresh and frozen samples. But this has now actually evolved to a clinical test that we call PAM50. Again, this is the original paper which first described the molecular portraits of human breast tumors. And I think most of you in the audience recognize how these work. You can make your RNA, change it to cDNA, and you put it down on a chip or a bead that already has prearranged probes that are fluorescently tagged or labeled that allows one to actually see whether or not you're getting hybridization. There's been an evolution in the PAM50. It was later adapted to quantitative PCR, but now uses something called nanostring, which again, probably many of you are familiar with. Rather than barcoding the actual individual molecules, now you're barcoding the actual probe with different light floors, and that allows one to have very, very small amounts of nucleic acid that can now be used for this analysis. So there's a huge advantage in that because many times our working substrate are small diagnostic tissue biopsies, and this allows direct binding without having to do preamplification. So this is really now a commercialized assay for breast cancer. One can actually order the PAM50 for breast tumors. It actually hasn't been that widely adopted because the usefulness of this has yet to be determined. One can actually do prognostic studies saying, oh, if you have this type of gene expression, you have so-and-so a prognosis. But in the business of oncology or the tenets of medicine in general are why order a test or really never order a test unless it's going to really change your clinical management. And because there's no predictive ability as of yet that's been proven, or at least definitively proven for many of these assays, most clinical oncologists have not actually ordered this on a routine basis. There's another test that's similar in this regard called MammaPrint, and again, this is another gene expression profiling. And again, it has the advantage now, although not originally, that it was unbiased. That it didn't require a priori knowledge of what was going on, that it was really used to categorize different types of breast cancers and then have some prognostic value, that this gene expression signature would be worse than this one and this one would be better than the other. It did not take into account receptor biology, which is something we always take into account nowadays, and it did require frozen tissue. Clinically, again, this has not been widely used because there are ongoing studies and some suggestion that this can have predictive ability, but it was kind of late to the game. It took a number of years before it could be adopted to the use of non-frozen tissue, which is not really what we do routinely in pathology for cancer. So it still does use the microarray technology, but can now be utilized on formalin-fixed paraffin-embedded specimens, and it's a 70 gene assay. Studies are really still ongoing to determine how good it's going to be for predictive ability for breast cancer. And then finally, Oncotype DX. This is, again, a gene expression test. By today's standards, it's a little bit archaic because it is quantitative real-time PCR, not RNA-seq. But this was a little bit different because this really addressed a problem that was very, very common amongst breast oncology, and that is early stage patients who are estrogen receptor positive. As a group, these patients generally don't seem to derive benefit from chemotherapy. And so it's always been a conundrum of breast oncologists after surgery, should this patient, who's clearly going to get some form of hormone therapy because they're estrogen receptor positive, should they get chemotherapy? And so that's really the central question that Oncotype set out to address. They also did something that I think was very smart of them to develop this going forward. They said, we're going to have to adopt this to an FFPE-based platform because that's how most clinical samples are going to come. And so we really got to figure out a way to extract good enough analyte to really do this on the vast majority of our patient samples. And the other thing they did was they really put the limelight on what we now call retrospective analysis of prospectively collected samples. So they partnered with various big oncology groups that had done seminal adjuvant therapy trials, meaning that these are patients who get additional chemotherapy or not after surgery, and they actually collected those samples. So the value is that these were all patients who were very controlled. They were in one arm versus the other. Again, they are retrospective, but at least the samples were collected prospectively. And what they proved using various training and validation sets is that Oncotype really has both prognostic and predictive value for breast cancer patients. This has been extended over the past few years to node-positive patients, particularly or exclusively really in postmenopausal node-positive patients, because that's the study that they based that off of. Again, I'm going to not go over this too much, but I'm sure everyone, or most everyone in this audience already knows about polymerase chain reaction, and this is just a way that one can selectively amplify a given region in a template DNA molecule, and it's exponential is really the point I wanted to make with this slide, but also as a foundation or a backbone for quantitative real-time PCR. This is still what Oncotype uses, and they do it extremely well. I've actually visited their facilities. It's all very tightly quality controlled, including all their machines and every lot of reagents that they get. But it employs simple TACMan probes, and for those of you who don't know, it's basically PCR. One requires a polymerase that has 5' to 3' exonuclease activity. So once a given probe binds to a region of cDNA, if it's bound there and then you get PCR amplification, that the TAC molecule will chew off the 5' end. And now this probe, which is normally not fluorescent because of this quencher in close proximity, gets liberated into the supernatant, and that's how you get your signal and noise. And this is how the readout is. The earlier your signal comes up, the more starting substrate one has. So Oncotype uses this technology, and they developed a 21 gene panel. There's 5 internal reference genes and 16 assay genes. They have their own kind of proprietary algorithm, and really this is the biggest paper that initially put it on the map. And again, this was a collaboration of both Genomic Health, who makes Oncotype, as well as several national adjuvant institutions which have done seminal research in clinical breast oncology for adjuvant therapies. One of the things that I always like to point out, however, is that not all of their test scores are the same. For example, the postmenopausal node-positive women, their recurrence scores and where they lie out in terms of prognosis is different because it was a different study. It's a different population. So again, one can't necessarily extrapolate that data into premenopausal women who are node-positive. So studies are ongoing. There are many prospective studies that have been launched. Or large prospective studies, I should say. One of them, which was launched a number of years ago, some early data has come out. And so the mature data on the question we really want to know is, and that is, what is the value of Oncotype for women who fall in the intermediate risk category? This is by far the vast majority of women in this country, and we don't really know what to do with that in terms of chemotherapy. The retrospective analysis didn't really pan out in terms of giving us enough confidence to say one way or the other, and that's why this prospective assay or prospective study was launched on this assay. So we don't have that data yet, but what we do have recently, this was just published a few weeks ago in the New England Journal of Medicine, is that they analyzed the low risk group. And sure enough, if you are low risk, you really don't need chemotherapy. So this is a prospective validation of that low risk population. This is kind of the score that you get, and this is what I mean by this, is that you have your kind of recurrence score, and this is the continuum. And so for this particular patient, it falls right there. And this will give you the readout at 5 or 10 years risk of recurrence or mortality. There are confidence intervals, as you see. I think the most useful part about this is what I was saying earlier, is the predictive ability. And so again, in this retrospective analysis of the prospective trials, one can see that if one has a low risk recurrence score, one doesn't really derive any benefit from chemotherapy, and the opposite is true for the high risk group. The intermediate risk group, although it doesn't look like they would derive benefit, the confidence intervals were big enough so that there was no way to definitively conclude that. All right, so a couple of other DNA tests that we can use and do use clinically. So DNA copy number is also something that we check for, particularly for breast cancer. Fluorescence in situ hybridization, or FISH, is something we do all the time for HER2. We'd like to know whether or not HER2 is amplified, because that actually opens up new therapies for that population of breast cancer patients. There are also SNP arrays, or single nucleotide polymorphism arrays. Again, this is still mostly in the research realm, as well as CGH, or comparative genomic hybridization. Gene sequencing is something I'm going to spend a little bit more time about, and this is something where we actually now have the capacity in a very fast way to understand or identify the different mutations in genomic and genetic alterations found in a patient's breast cancer. And so again, there's next generation sequencing, which I'm again going to mention a little bit later on, which one can do DNA sequencing of the tumor, one can do RNA-seq, as we mentioned, so it's essentially the same thing, but converting the RNA to cDNA. And there's still other methodologies that are still in the research realm, ChIP-seq, methyl-seq, etc. So again, FISH, for those of you who don't know, this is really a old school technique by today's standard, and really what one does is develop these probes that are fluorescently labeled, and in situ will actually hybridize to a given region of a cell's genome. And so the nice thing about FISH is you can get these beautiful pictures looking at different fluorescent probes and do copy number analysis. The bad thing about FISH is that, although I think many people think this is a very quantitative and highly reproducible type of assay, and it does have good reproducibility, but it is something that is an art. If one has ever done FISH, you need to know how to count the cells, how to focus in and out, because the nuclei can get shaved in half. And so there is a low level of inherent noise. And so FISH is not a perfect science by any means. It is a very good test. And again, there are parameters on how pathologists and cytogeneticists can count these dots to really say if someone's HER2 positive or HER2 negative. Again, SNP arrays, I'm not going to talk too much about this other than to say that this is still an ongoing research tool. SNP arrays were originally used, just as the name suggests, to identify variations between maternal and paternal alleles within an individual. With clever bioinformatics, people actually figured out that one can use this in cancer for many different purposes, including copy number variations and deletions, and so as well as loss of heterozygosity analysis. And so again, even though this hasn't taken off in the clinical realm, this is something that is still being used by many breast cancer and other cancer laboratories throughout the world. How does this actually work? Again, it's very akin to the microarrays that I showed earlier. If one looks at various single nucleotide polymorphisms as depicted here, many times one has homozygosity, in this case of an intron between a 3-exon gene, A and A, and many times one is heterozygous because, again, one allele is from mom and one is from dad. By putting these different bits of DNA onto a chip or a bead array that has fluorescently labeled probes that are specific for one or the other, one can then identify what the SNPs are in a given locus. And so again, the utilities, as I mentioned earlier, have really spread. I think the classic use of these were GWAS studies, or genome-wide association studies, which are, in my view, still correlative, not causative. And as I mentioned before, there are a number of useful parameters that we can analyze using these SNP arrays for cancer. And now DNA sequencing. So DNA sequencing, I think, again, most people know what this is. This is simply the ability to decode what's in a patient's genome. The first generation, as I like to call it, was really Sanger sequencing and pyrosequencing. This is still used quite a bit, both in regular clinical oncology, but also in the research lab. It's cheap, it's fast nowadays. It's nothing like in the old days. I have to show my students pictures of gels to make them understand how difficult simple sequencing used to be in the old days. And then came along next generation sequencing. Really the first one, which was a little bit forgotten now, was called 454. Then came ion torrent, as well as solid platforms with Illumina, etc. And how is this done? Just as a refresher, again, in the old days, what we would do is mix up regular old DNTPs, ACG and T, with dideoxys. And in the old days, these were radioactively labeled. And so a dideoxy will cause chain termination. And one can actually then see how this works. As you get incorporation of a false nucleotide, it stops, but others will still go until they incorporate the false nucleotide and so on. And because there was no distinction between radionucleotides back then, we had to run 4 lanes for every sample, a G, C, A, and T, and one would simply go up the ladder to get the sequence, A, T, A, and so forth. The big kind of breakthrough was that we could then label, rather than the radioactivity, we could label individual fluorescent probes or molecules for the dideoxys. And so now G, A, T, and C all have their distinct color. Rather than running 4 lanes, one could actually do it in a single lane. And rather than doing it in a gel, one could actually do it in a capillary that had gel filled with it. This greatly increased the cadence and the pace of how one could do sequencing, and actually now is the basis for most Sanger sequencing done. This is just from our own work, where you can see how this would help us in the clinical realm as well as the research realm, is that when you actually PCR amplify a given locus, in this case a very commonly mutated area of the KRAS gene, if one has a mutation and it's a pure tumor sample, you should see the 50-50 distribution of that particular mutation as opposed to in the wild type controls. Now, as great as and easy as that is, Sanger sequencing has a number of limitations, chief among them being that it doesn't assay for individual DNA molecules. It assays for a summation, generally speaking. And when that happens, you can actually miss low-level mutations, generally speaking, because if you have a summation at any given nucleotide, that means you have to have probably at least 15 to 20% of your mutation present. Otherwise, it'll be drowned out by the wild-type signal. And so next-generation sequencing really actually kind of tackled a number of different problems, but probably the 2 biggest ones are, one, can we do this faster and more globally? Can we sequence the whole genome in a much faster way? And then 2, can we improve the sensitivity of low allelic abundance, meaning that if your mutation is only present in a small percentage, can we actually now detect it? So again, for those of you who don't know, next-gen sequencing, 1, relies upon that we have a reference human genome, and then 2 is that we chop up the DNA into little fragments and then sequence one or both ends. And we have to do this redundantly to make sure that we actually get what's called enough depth of coverage to feel confident that if we see a mutation, it's present there multiple times. That's really the brilliance of next-gen sequencing, is that we're doing everything massively in parallel. And so the idea about how it sequences is relatively the same, but the innovation really is that now we're doing this, again, multiple times massively in parallel. So NGS, we can now sequence billions of base pairs of DNA in a time and cost-efficient manner. It does require redundant coverage, as I said before, and we can detect rare mutant DNA populations now with better bioinformatics tools at really about 1% allelic frequency or even less. We also have flexibility to not do the whole genome. We can actually capture regions of interest. There are 2 different ways that really one can do this. One can PCR amplify a given locus or a set of loci. Probably the more common methodology is actually to have biotinnylated probes. And if this is a whole exome sequencing, let's say, for example, the orange bars represent exonic sequences, one then takes the sheared DNA from a genomic DNA source and then hybridizes this and then pulls them out by virtue of this biotinnylated probes with streptavidin and magnetic beads, and you've effectively captured just the regions you want to do sequencing on. This has the advantage because rather than now doing 3 billion base pairs, which is the haploid human genome, one can only, for whole exome, let's say 50 million base pairs. So orders of magnitude difference, so you can actually get more bang for your buck in a sequencing run. The other advantage, it requires very little starting material. You can actually electronically figure out surrounding normal cells, and they can be electronically accounted for. And this is very critical for what we do in cancer, because many times we have these FFPE slides, and there's going to be surrounding infiltrating normal cells both in and around the tumor. We can sometimes also get very good information for copy number and loss of heterozygosity. We can identify rearrangements such as translocations. And probably the thing that's the biggest focus and how it's actually going to impact clinical oncology care is, can we identify specific areas right now in 2015 that have drugs if a gene is mutated? And so rather than doing, again, a whole genome or even a whole exome approach, one can just target and pull out genes that are actually clinically relevant for oncology today. There are challenges, of course. It's still does require some cost and time, particularly the bioinformatics end. Again, you can imagine having billions of base pairs and putting them through a computer and then figuring out what's what first. You have to align it against the human genome, and then what's different can be challenging, to say the least. Again, whole genome and exome approaches may require more input DNA, although improvements continue to occur in this area. Variant calling is probably, I think, the biggest challenge of this field, and that again is the bioinformatics side. There have been studies that if one takes the same raw data and puts it through various variant callers, maybe at best you'll get 50% concordance. And again, I think the field is really tackling this problem to move to a more unified approach. But this is really something where we struggle with every day with in the bioinformatics field and in the cancer genetics field in general. How do we really know whether this is a true mutation or not? And again, I'm not going to spend too much time other than RNA-seq is probably the new way to do gene expression analysis, because one can do a global analysis much more readily as opposed to the old ways of quantitative real-time PCR. Foundation One is probably one of the first tests out there put out by a company called Foundation Medicine, and this is actually commercially available for all cancer types, and one can send off a tumor, and basically it's the FFPE block or slides cut from the block, and they will extract out the DNA and perform next-gen sequencing on a panel of cancer genes. It has very high sensitivity and specificity. It's CLIA-grade data, which is necessary if we're going to make clinical recommendations off this data. And again, they were one of the first to really use a cancer gene panel approach rather than a whole exome or whole genome approach. That actually had several big implications. One, the turnaround test was very quick, because rather than doing, again, all the different genes in the human transcriptome, they were focusing just initially on a panel of about 100 genes or so. So the bioinformatics challenge was not as great. Secondly, it also allowed them for a given sequencing run to have much higher depth of coverage. Prior to this approach, people were doing whole exome sequencing maybe with 10 to 100-fold coverage, meaning for every given loci, you were only getting about 10 or 100 reads. Although that probably is good enough for many circumstances, in some situations it's not. There are inherent artifacts of next-gen sequencing and PCR artifacts that preclude confidence if you only see a mutation once or twice. And so having at least 500x coverage minimally for any given locus was kind of their goal. And so that gave a lot more confidence that if they reported out a mutation, in fact, that was real. And this has really been widely adopted for many commercial entities and even academic institutions now as the approach moving forward in terms of what is a good cancer gene next-gen sequencing approach. So again, this is just the conclusion of my talk before I get into the gateway tumor board, but hopefully I've walked you through this, that molecular genetics and genomic approaches have really increased our understanding of breast cancer and our ability to make clinical decisions regarding risk, therapies, and prognosis. Again, this is probably true mostly for the oncotype test, but these other assays are quickly catching up. The use of these assays that measure protein, DNA, and RNA in particular have revealed a lot of new insights, and particularly the complexities of breast cancer. That's probably been the most eye-opening and somewhat sobering discoveries of the past decade, that it seems like every breast cancer patient is going to be a little bit different from one another, and some are vastly different from one another. So again, I think there's now this idea and notion put forth by cancer centers everywhere, President Obama with the precision medicine initiative, that a one-size-fits-all approach really isn't going to work for a disease like cancer, and breast cancer specifically. So I'm going to shift gears in the last 15 or so minutes about something that we started back in, I guess 2012 now. And this is something that I came up with this acronym because I was applying for a grant, and I thought it would be good to have a catchy phrase for our moniker for our tumor board. Turns out I didn't get the grant, so I don't think it really worked. But anyways, the name of our tumor board, it's a molecular profile tumor board, is the Genetic Alterations in Tumors with Actionable Yields, or Gateway. I thought that was a little bit punny, so that's the name that I went with. It originally started with a number of colleagues, and currently Josh Loring, who's also in the breast cancer group, and I really are the co-directors and lead this on a weekly basis. So for any of you who do have any reports who are clinicians that you don't know what to do with this, we welcome, if you want to submit your case, just send me an email and I'd be happy to look it over and present it to our tumor board. What is our purpose? Our cancer center, again, about 3 or so years ago, launched a personalized medicine effort. And so we're gearing up to try to have every patient who walks through the door, their cancer genomically profiled with next-gen sequencing, to have that information stored in a database, to have it eventually reported out into their electronic patient medical record, and also to have that information de-identified for research purposes, because it will also be tied to clinical information. Part of the problem that was emerging during this time, because this was also when companies started putting this out, is that the information one got on the sheet just said, oh, if you have this mutation, you should consider this type of therapy. And that wasn't necessarily true. There were some leaps of faith that were being made by companies as well as the people who were interpreting this. And many parts of the world and many parts of this country, lots of clinical oncologists don't have a genetics background. They don't have training into understanding that not all mutations are the same, or that even a mutation that's the same in one cancer type may not respond in a different cancer type. So clearly there was an unmet need in how to interpret these genomic profiling reports, and that's really what the genesis of our tumor board was. So the mission, again, there's a lot of writing here. I'm just going to paraphrase a lot of this stuff. But really, we want to review a patient's molecular tumor profile. We're not limited to any one platform, and we're certainly not limited to any one type of analysis. That is, we'll look at things that have mutations, copy number changes, sometimes even IHC, because they're all on the same report. And we'll do it from any source, whether it's academic or commercial. What our job really is to do, and again, I have to thank my team for doing this. It's all done on a volunteer basis, but we want to review the relevant literature. And so, as you might imagine, things have actually changed in our recommendations from 2015 versus 2012. But we want to go over the level of evidence and see, well, is there any evidence, and how weak or how strong is it that this particular mutation would actually predict for sensitivity to a given drug. We also review the current state of FDA-approved therapies and clinical trials. And we really, again, try to figure out what is truly actionable versus what is not likely to be very actionable. We discuss this at our tumor board. Again, it meets on Mondays at 11. And we actually write up a nice summation and give it back to the referring physician and the patient. So depending on what you read and who you read it from, maybe even what your religion is, actionable is really defined many different ways. We're kind of sticking with these 4 bullet points, and this is something that I think a lot of tumor boards have also adopted, that the first actionable definition of an actionable mutation would be a genetic alteration that already has an FDA-approved therapy. So for those of you who don't know, about 50% of melanomas will actually have a mutation, a very specific mutation in the BRAF gene, and that's V600E, and we have now a couple of different drugs that can actually target that mutation. The second idea would be a genetic alteration that has the same approved therapy, but it's in a different cancer type. And again, one could extrapolate from vemurafenib data in metastatic melanoma that this might also work for other cancers that have that V600E BRAF mutation. Turns out it's not that simple. For sure, there are other cancers that respond to these drugs that have this mutation, but as probably a most notable example, in fact, it was just published in Journal of Clinical Oncology, that in fact, colorectal cancers that have this mutation do not really respond to vemurafenib. And there have been a number of big papers in Nature and other journals that have suggested why that might be, and that's led to kind of rationally designed trials targeting 2 different pathways and not just the V600E mutation. The third definition would be a genetic alteration that may provide rationale for participation in a clinical trial of a targeted therapy. And then finally, a genetic alteration, and we kind of added this, but I think companies and institutions have adopted this, a genetic alteration that may lead to recommendations for genetic counseling and germline mutation testing. Many of these companies actually do not do in parallel normal control germline from the same individual. And that has both advantages and disadvantages. Obviously, the biggest disadvantage is that if you only do the tumor, you may actually be picking out things that are in the germline, and so they may be not really relevant to that tumor as a somatic change. A disadvantage, however, and we've actually found this to be the case in many of our tumor boards, is that if the clinical context is right, let's say it's a young woman who actually has a strong family history, who ended up having breast cancer, if we find in the tumor a p53 mutation or BRCA1 or 2 mutation, that actually could be in the germline, and that would actually get filtered out by most strategies doing sequencing of both unless specifically asked for. And so in our view, that has actually been very helpful because we have actually made a number of recommendations in the appropriate clinical context to refer to a genetic counselor and get the real counseling and bona fide genetic testing if deemed appropriate. Additional considerations, I think one of the things that it's easy to do if one doesn't see the patient is just to say, oh, we've got the data, and so here are the mutations, and this is what we would recommend. It's much more difficult to understand in the clinical context, is this patient on their first-line therapy, their fourth-line therapy, et cetera, and how does that weigh in compared to taking the chance, if you enroll this trial, that it may or may not be effective, or take this drug that may or may not be effective. Discovering and interpreting potential germline variants, again, and the need for genetic testing counseling is something that we've really factored in from day one. And so we actually do have Jen Axelman, who's a certified genetic counselor on our tumor board, and she has been wonderful in helping us with these issues. And then finally, legal and even ethical implications. Aril Chennai's group at the University of Michigan, they first started doing this with whole genome sequencing. One of the things they actually found in a prostate cancer patient was proviral HIV DNA integrated into the DNA of that patient's probably T cells, but they actually found it in the actual sampling of the tumor. And so that individual didn't know he had HIV. And so what are the legal ethical implications of that? And so from the very beginning, these were the charter members, many of them are still here. We had a number of experts and expertise outside the realm of cancer genetics and medical oncology, and I'm not going to go through all of them, but again, I think, as I mentioned, we also have Jen Axelman, who's our genetic counselor, we have Cindy Gagan, who's a patient advocate, and we have ad hoc representation, if we need it, from the Berman Institute with Jeremy Sugarman in ethics and Meg Garrett in legal. I think this has been really helpful for us to say going forward that we're trying to anticipate the unanticipated. This has also been useful for our own internal study in breast cancer, looking at whether or not we can use this concept of what we call liquid biopsy. But really the primary objective of this particular study was to see whether or not we could get a fresh biopsy and take it all the way in 28 days from getting that biopsy to getting it sequenced. Foundation Medicine did our sequencing for us, and then actually convening the tumor board, and if we could do that in a 28-day time frame. That time frame was chosen specifically because often that's the washout period a patient is going to undergo before starting a new therapy. And so this was the patient characteristics. There was a total of 23 patients. We are still enrolling a kind of an appendix trial right now, looking at the feasibility of using specialized blood collection tubes. But for the most part, this study is done, and it was done specifically in triple-negative breast cancer patients because they have, in breast cancer, limited options in terms of systemic therapies. Really, right now, chemotherapy is the only standard of care. And we don't get a lot, quite honestly. When we convene our tumor board, we get a lot of information, but in terms of things that are really actionable, that's where it becomes much more difficult. And other centers have since published similar data stating that a big impediment is, even if you have something that makes sense for a clinical trial, that trial is often, in a different part of the world, and patients really don't have the financial means to just pick up everything and leave and move. And so again, what is really actionable becomes the key question, and that's something I think the field is really trying to figure out. We did have a couple of cases, though, where we found in one case something that was interesting because it was an amplification of MEK. MEP2K1 is the Hugo name. And so there is a drug that targets this. It's actually approved in melanoma in conjunction with one of these mutant-specific BRAF inhibitors. It was not our first recommendation. We said, how about a trial of targeting of a MEK inhibitor? But again, not being available, the referring physician ended up getting approval and putting this patient on trametinib. And we had follow-up that, in fact, she had a big chest wall lesion that actually responded very nicely to this drug. And what we've been kind of touting is that really the most rational way to think about this is that if the drug actually hits the target or is maybe one step above or below in the pathway, that's probably got a higher likelihood of working. And that's kind of an emerging theme, I think, that's been coming out of the cancer genetics and clinical oncology data. We had a second patient, which is very interesting because she actually had a mutation in c-KIT, and this is a mutation that is found in GI stromal tumors, or GIST. That's important and relevant because GIST tumors actually have a drug. It's called Gleevec, or imatinib. It was first developed against CML, against BCR-ABL, but it also has remarkable effectiveness against certain types of mutations in KIT found in these tumors. So we were actually very surprised to see this because we had never seen this before. We did get approval for off-label use of imatinib to treat this patient. She really didn't have any treatment options. I was actually the inpatient attending when we got these results, and she was hospitalized. She actually had low-level bacteremia, really had burned out on all our therapies. So there wasn't really much we could do for her in terms of standard of care therapies. We got approval, and unfortunately she passed away in 5 days after we started the drug, so we don't really know whether she would have responded or not. But that was one of the few instances that we had a real mutation approved in a different type of cancer that we actually found in breast cancer, or a different disease state. So again, the conclusions of this portion of the talk is that the Gateway Tumor Board is operational, and again, we are available if people need our help. I feel that it does add values to the Hopkins personalized medicine effort, and probably the personalized medicine effort just globally for cancer genome sequencing. This really, I think, is the missing link that is going to be necessary for many institutes and companies as they move forward, of how you actually parlay what you find in the tumor into something that's going to hopefully be of clinical benefit for the patient and the referring physician. We'll continue to follow up on our physicians. We have probably well over like 150 cases at this point, and we are going to write this up, and we're following what the outcomes were of these patients from our reports. And with that, I think I am done, so I'd be happy to welcome any questions if there are any.
Thank you. We have a new microphone set up, so we'll bring the microphone to you. Just raise your hand with your question.
Thank you, Ben. So going back to the Boltstein data set, these 4 major types of breast cancer, now in current day knowledge, how useful was that? And to what degree is there heterogeneity, and is there any use to classifying these breast tumors into major categories?
It's unclear how useful it is right now. We throw around the lingo, but clinically, we don't really distinguish right now from luminal A or luminal B. We treat them still with ER-positive therapies. Some of us might be more inclined to treat chemotherapy if it looks like a luminal B. And I think there's a number of studies out there that use kind of immunohistochemistry as a surrogate marker for luminal B, but they don't necessarily actually send out the test to prove that it's luminal B. And so right now, you're absolutely right. There's not been a lot of clinical utility for categorizing by gene expression how we treat patients. Really, there's none, I would say. I think we still use the standard ER, PR, HER2, IHC staining, immunohistochemical staining, and in cases of HER2 equivocal staining, we will then reflex to FISH testing. And that really still remains the standard of care. I think that could change moving forward. Jennifer Pietenpaul's group at Vanderbilt University actually had a paper a few years ago doing a similar type of strategy of gene expression profiling on just triple negatives, and showing that even within the triple negative population, you can have varying different subgroups. So there's at least probably 6 different subgroups of triple negative. Clinically, we've seen this before. For whatever reason, we would see occasionally a patient who has very slow indolent disease despite being triple negative. The majority are what Chuck Peru's classification would call basal. But again, there is a subtype that is, for example, triple negative but expresses androgen receptor. Those tend to be now considered triple negative luminal types, or luminal AR, and they tend to behave more like ER positive breast cancers in that they're slower growing. There are trials looking at whether we can target AR in that population, androgen receptor. So again, I think it's become a good platform for clinical trials to see whether or not there is going to be clinical utility of doing some sort of gene expression profiling and managing our patients going forward.
Just pushing the argument a bit. So you have a long-standing experience in breast cancer. So from comparing the usefulness of this molecular data to just looking at a biopsy, what do you feel better about in terms of knowing about prognosis and the response to treatment? Just looking at the biopsy phenotype. Yeah. Might that be better still?
Yeah, it is still the standard of care. For breast cancer, and again, I think a lot of people have maybe misinterpreted my involvement with the Gateway Tumor Board. The reason I got elected to lead this was I was having a discussion with our now Deputy Dean Liz Jaffe, who's heading up this effort as the Associate Director of Translational Research. And I was pointing out to her a couple of years ago of a report that I saw from a company that suggested this patient had a PIK3CA mutation and should go on a PI3 kinase inhibitor trial, and she would have to At the time, it was only available in Boston, so she was packing up her bags and she was going to go there. And she had metastatic incurable breast cancer that she hadn't really received standard of care therapies yet, with proven value of extending life, of improving mortality. And again, that's within a population. But she went ahead anyways. My issue with this was that PIK3CA mutation, it was not known to really be a functional consequence. It wasn't what we call the hotspot mutation. It could have just been a variant. But she went up there, she went on the trial, she had a horrible reaction. She was intubated in the ICU for several weeks, and because of her young age and reserve, probably got off and was okay from that particular episode. But that really struck a nerve with me, and I felt like, we really need to counsel patients that they are foregoing potential life-prolonging therapies for this shot in the dark. And that's when Liz said, Ben, you've clearly thought about this. You should really lead this effort. So I tend to be much more conservative than I think a lot of cancer centers that are doing translational research types of molecular tumor boards because of these types of experiences. Having said that, when that KIT mutation came up in that patient, we had a long debate in our tumor board, and many of our tumor board, particularly those who are more clinically inclined, didn't think we should try this. And in my view, that's exactly the gold mine that we're looking for when you actually have a mutation that clearly has benefit in another tumor type that has a drug that's FDA approved for that tumor type. Now, again, there's no guarantee that would work, and I just pointed out that vemurafenib does not work in BRAF mutant colorectal cancers, but to me, that woman did not have any other options, and this is a very relatively safe and well-tolerated drug. It's a very expensive drug, and that's a whole nother discussion, but that's again, something that I felt we really uncovered that wouldn't have been uncovered by standard IHC.
I had a question. It might be a little outside of your sphere of interest. So we're interested in etiology and cause. I'm wondering if there are any patterns of mutational spectrum that are involved, say, breast cancer, that seem to be related to factors like premenopausal, postmenopausal, smoking, non-smoking. Obviously, they're familial markers, but I'm trying to think of others.
It is a great question, and the truthful answer is that we don't know that yet 100%. There have been some studies looking at different types of newfound genomic instability based upon certain types of genes, but again, even that work is still early on. I think what we have learned, though, is that with the different subtypes of breast cancer also come different mutational spectra that are more likely to occur in one type versus the other. As an example, the luminal breast cancers, or the ER positives, they tend to have about a 50% rate of PIK3CA mutations, whereas triple negatives probably only really have about less than 10%. And when you look at Jennifer Pietenpaul's data, it seems like the 10% probably are enriching for those triple negatives that have androgen receptor expression. So again, for whatever reason, it looks like PIK3CA mutations tend to be more associated with breast cancers that have some sort of steroid hormone receptor expression. Conversely, when one looks across the mutational landscape of triple negative breast cancers, P53 mutations are through the roof. And in fact, there was an earlier British Columbia study in Nature that suggested maybe only 50%, but the TCGA data came out, suggested it's as high as 80 to 90%. And our own data of triple negative breast cancer patients, it was every single patient. Now, granted, that was only 20 plus some patients, but it is pretty striking. And in fact, I've kind of put forward the idea that P53 for triple negative breast cancer patients may be the initiating driver, similar to what APC is for colorectal cancer. So I do think there's the substrate there, the foundation to really data mine this and figure those types of questions out. And I think we haven't gone deep enough. The other thing that has also emerged is that TCGA is largely just primary tumors. And so about 2 years ago, a number of groups came out with papers suggesting, in fact, proving that hormone therapy resistance is associated with receptors in the estrogen receptor, or mutations in the estrogen receptor. That seems so obvious, and yet as a field we hadn't really done a lot of metastatic biopsies, and so that really delayed the discovery of this. In fact, it's interesting, similar to many other drug-resistant stories, there are a number of groups 2 decades ago that actually proved in vitro and also in xenograft models that those mutations existed, and people just were kind of, well, it's not real human data, so we don't know what to make of it. And then 20 years later, in fact, it was proven to be the case. So there are opportunities to actually figure out whether there are certain mutational spectra, whether or not that's going to be associated, or one can figure that out, whether there's additional environmental insults. I don't know. I've always said it's really tough, and I'm sure you know this as well, because breast cancer is so common. Average risk is 1 in 8 women, and that's clearly going to be related to estrogen exposure. To define any additional insult or threat that contributes to breast carcinogenesis becomes very difficult, and I would suspect that's also going to be true at the mutational level.
So then a question for me. How have the patients received this genetic information?
Yeah, so a variety of different ways. I think many times what ends up happening is that the referring physician or one of us will actually, who is the referring physician on the tumor board, will actually relay this information back to the patient. One of the things that I try to do is temper, when the test is being ordered, temper the expectations to say, this is not a magic bullet. We're not necessarily or not likely to really find anything that's going to be truly actionable, meaning that there'll be an off-the-shelf drug that we can order tomorrow. Most of the times what ends up happening, it provides some rationale for a clinical trial. And then the caveats is what I said earlier, that trial may be in Timbuktu and not accessible for the patient. So again, I think there are a lot of challenges of not giving false hope and false expectation to patients, and I think that's again hopefully something that we can also help put out to the public as well as to physicians. But this is really a challenging time because, in fact, the patient that I mentioned with that GIST mutation, the KIT mutation, while I was the inpatient attending, she said, I'm waiting for this test because I'm just sure I'm going to find something. And I tried to really squash her hopes, and I said, I lead this tumor board. We have yet to really find anything like that. I'm telling you, we're really, don't count on this. And the next morning I had to eat my own words because I couldn't believe it just came that night. I did a double take. I couldn't believe there was actually this mutation. So I think that's but that does highlight the fact that the majority of the times we don't get something like that.
Let's give Ben a hand. Thanks so much.
Thank you.